Thank you very much for the nice introduction and the kind invitation. It's a pleasure to be back in Paris. And it's also very stimulating to see this meeting where researchers and clinicians from all over the country come to meet. It's a nice network, something to be jealous of. So I will be talking about antisense oligonucleotide therapies for ALS. These are some of my disclosures. As we all know, finding a cure for ALS has proven to be a difficult thing. We have had many negative trials over the last 3 decades. And one of the reasons for this could be that ALS is a heterogeneous disorder. It's a heterogeneous disorder at the clinical level. The disease presentation is variable. At what age it starts is variable. How quickly it progresses is variable. But I think these parameters, this heterogeneity in clinical parameters is something we should be able to address with better clinical trial design. More troublesome in my mind is the variability in the underlying biology of ALS. And so not only the causes of the disease are heterogeneous, there are now more than 40 genes associated with ALS, but also the underlying disease mechanisms are variable. And there are many different things that go wrong in motor neurons and in glial cells, and we don't really know which of these pathways are important for which patients. And so this cartoon depicts a bit the same problem. ALS as a whole, there are many different subtypes of the disease. ALS is probably rather a syndrome and maybe not a single disease. And therefore, a single treatment may not exist. And an obvious solution to this problem is precision medicine, and that is emerging. For genetic subtypes, it is possible to just study that subtype of the disease, and this has been proven to be a very powerful approach. And we now know that, for example, patients with the SOT1 mutation and patients with the FUS mutation probably have quite a distinct form of ALS that should be treated differently. Having said that, the neuropathology of ALS is much less heterogeneous. 97% has TDP pathology, with TDP moving out of the nucleus, aggregating in the cytoplasm, which can cause nuclear loss of function and cytoplasmic gain of function. But as of today, it's not entirely clear how these two mechanisms give rise to motor neuron degeneration. And so when you think of therapeutic strategies for ALS, you can use 2 approaches. You can try to target the upstream disease cause, if you know the gene mutation that is causing the disease, or, and that's the case for the majority of patients, you can try to interfere with the downstream disease mechanisms if you don't know the cause. There is a lot of enthusiasm in the field around antisense oligonucleotide therapies because of the recent successes, and this method has several advantages. It allows for a precise intervention. You can modulate the expression of a certain gene. You can correct splice errors. It's even possible now to increase expression of genes. For instance, if you design an ASO that binds to upstream open reading frames. And these antisense oligonucleotides, as they interact by specific base pairing, they have few off-target effects. They are modified so that they have a very long half-life compared to drugs. They have a very good penetration in tissue, in the CNS tissue. Another very important advantage for early clinical trials is that you can give variable doses. You can start low and go higher. It can be stopped when there are side effects. But the major disadvantage, of course, is that it does not cross the blood-brain barrier, so you have to administer it intrathecally. What I would like to do now is give a brief overview of the ASO studies that have been done and that are ongoing. And this is a figure from a recent review we made in Trends in Molecular Medicine. And there we group them, the ASOs, in different categories. You can try to tackle the genetic cause of the disease. It's not gene therapy. You don't correct the gene mutation, but you can, for instance, silence the gene product. You can also tackle genetic risk factors of the disease. also splice defects that are related to a loss of TDP function, but basically any pathogenic mechanism you think is relevant for ALS, you can try to tackle with ASO therapy. And this means, I think, nowadays that observations in the lab, when you find robust therapeutic targets, it is possible to very quickly translate that into a therapy that can be tested in a phase 1 biomarker-guided trial in patients. So let me first start with some of the progress that has been made in the genetic subtypes of the disease. SOT1 ALS, we all know it very well. The mutations are mostly missense mutations. They are relatively common, up to 20% of familial ALS in total, 1 to 2% of cases. So it's only a small proportion of the total ALS population. The phenotype associated with ALS and with SOT1 mutations is variable, but on average it starts earlier. Oftentimes it's a lower limb onset, a lot of lower motor neuron involvement, although some forms with prominent upper motor neuron involvement exist as well. And depending on the mutation, the phenotype can be slow or fast. The disease mechanism is a toxic gain of function rather than a loss of the oxide dismutase activity. And here you see one of these cartoons depicting all the different mechanisms that occur downstream of the SOT1 mutation. But in the mice, it became evident that simply reducing the transgene is a very powerful method to treat the disease. And so this gave rise to the development of ASOs that bind to the SOT1 mRNA and reduce SOT1 protein levels in a non-allele specific manner. So both the wild type and the mutant protein are reduced. And this method was first tested in mice by Tim Miller and colleagues and Emma Campbell in Don Cleveland's lab. And the ASO worked well. It could reduce the SOT1 protein not dramatically, by 25%, but this gives rise to an extension of the lifespan in mice. And these results later triggered phase 1 trials in patients. And already in the phase 1 trial, it became evident that a dose of 100 milligram delivered once monthly gives rise to a reduction in SOT1 protein levels, but also in neurofilament levels, which is a marker of neurodegeneration. And this was largely confirmed in the phase 3 trial, where you see that monthly injections of tofersen give rise to about a 40% reduction of SOD1 levels in the CSF. This is quickly followed by a dramatic lowering of neurofilament levels by 50%. But the primary endpoint of the trial was after 6 months, and at that moment in time, although you see that the clinical evolution, the functional decline measured by the ALS-FRSR is slightly better in patients treated with tofersen, it doesn't reach statistical significance after 6 months. And the same is true for a mega score for the muscle strength. Patients on tofersen do a little bit better, but it's not statistically significant after 6 months. However, the long-term follow-up data clearly show convincing clinical response. Patients tend to stabilize, and they do much better than patients that received tofersen only after 6 months, and that difference is statistically significant. The muscle strength also seems to stabilize, and again, patients that started early on tofersen do better than the delayed starters. So this study is really a game changer for the field. I think there's the two most important conclusions are that one, of course, that tofersen is an effective therapy, and so this subtype of ALS becomes a treatable disease. But another important message, I think, in my mind, is that even when you have an intervention that has a large effect on the neurodegenerative process with a dramatic reduction of neurofilaments, it takes time before it becomes clinically evident. It takes 9 to 12 months before you can measure it at the clinical level. The second therapy is under development for FUS ALS. That's a rarer form of familial ALS, responsible for 1 to 3% of patients with familial ALS. There are some distinct phenotypes related to FUS mutations. Of course, the P525L mutation can give rise to a very aggressive juvenile form of the disease, but also the other mutations typically have a relatively early age of onset, a fast progression, and the onset can involve prominent neck extensor weakness and also proximal lower limb weakness, which is quite unusual in ALS. And typically the patients have prominent lower motor neuron involvement. The mutations are clustered in the C-terminal region, and the mechanisms are not fully understood, but it's just STDP, a DNA and RNA binding protein, and mutant FUS has a tendency to mislocalize to the cytoplasm where it can aggregate, but also affect gene expression and splicing of genes. For this form of ALS, an ASO was developed by Ionis in collaboration with the Don Cleveland Lab and Sandrine Cruz, and that was later tested by Neil Snyder's group. And here you see an example of the downregulation of FUS in different brain and spinal cord tissues. So the ASO seems to be very efficient in downregulating the expression of FUS protein. And this again is non-allele specific. It downregulates, it's intended to downregulate both wild type and mutant FUS. In the US, they have this expanded access program where patients can receive experimental therapies early on, and this allowed a rapid drug development process. Here you see the ALSFRS of a patient with juvenile ALS caused by a P525L mutation that was treated with increasing doses of the ASO and ended up in a quite stable, prolonged, presumably prolonged disease course before dying. But at autopsy, it was clear that the ASO is doing its job. The FUS protein and also the mutant version of the protein are almost completely wiped out. So the ASO is doing what it is supposed to do. More patients were treated in this program, and there is anecdotal evidence that there is reductions of neurofilaments in them, and even some patients that improve. So this led then to the initiation of a larger phase 3 study, which is currently ongoing, and of course the results are anxiously awaited. But potentially there is a second ASO therapy that can be helping patients with this specific subtype of ALS. The third genetic subtype that has been investigated is the C9, ALS and FTD. There, the efforts were less successful. The mutation is very common, as we all know. It causes FTD, but also ALS. And in the ALS phenotypes, it more commonly gives a bulbar onset. FTD is frequent, and typically there can be a faster disease progression, especially in males, although other phenotypes exist. The mechanism is not entirely clear. There is a reduction of the protein that can contribute. The protein plays a role in autophagosome maturation and autophagy. Reduced levels of the protein, especially in microglia, can be important. But there are also toxic gain of function mechanisms with the repeat RNA that can cause depletion of RNA binding proteins, and also the production of DPRs, which is further explained here. This is an overview of the disease mechanisms related to C9 ALS, and in particular, the sense RNA and the antisense RNA that contain the repeat. This is normally in an intronic sequence, and after splicing, these lariats are degraded, but as they are so GC-rich, they can form secondary structures, G quadruplexes, for instance, which can be translated to the cytoplasm, ribosomes can dock, and this can give rise to translation into protein, which is not meant to be there. Dipeptide repeat proteins, and so from the sense strand you can get poly-GP, GR, and GA, and from the antisense strand poly-GP, PR, and PA. And these DPRs, they are very prone to aggregation and can cause toxicity. They can disturb mitochondrial function, axonal transport, translation, and so on. So here the design for a therapeutic approach was to make an ASO directed against the two transcripts that contain the RNA, the sense transcript, but not touching the V2 transcript that does not contain the repeat, and so the production of full-length C9 protein is still possible. Two companies used this approach, Biogen and Wave, and the results of the Biogen study have recently been published. And as you can see, the ASO is beautifully doing what it is designed to do. Here you see a nice dose-dependent reduction of one of these DPRs, GP, to about 50%. So the drug is indeed reducing the production of these DPRs, which is very important. But unfortunately, we would all have hoped that this would also lower the neurofilament levels in patients, but here you see the neurofilament levels in controls, which remains relatively stable, but in patients it tended to increase by 25%. Nobody understands why. But also the clinical response seemed to be rather unfavorable. Here you see the placebo, they are going down, but the ones on ASO appear to be going down faster. And so this was the reason why the program was terminated. We don't really know why this approach did not work, but one possible explanation could be that if you block the sense transcript, maybe the antisense transcript is produced more, and from the antisense transcript we know that other DPRs are being formed, such as PR, which is one of the most toxic ones. But of course this remains pure speculation. Then our fourth big gene in ALS is STAR-DPP, the gene encoding TDP. It's also a rarer form of ALS, 1 to 5% of familial ALS. It typically also gives an earlier onset. Most commonly it starts in the upper limbs, bulbar onset is uncommon, and it typically has a slow disease progression. The mutations cluster in the C-terminal domain. And in terms of disease mechanisms, both toxic gain of function by cytoplasmic aggregation and nuclear loss of function have been implicated. But TDP also is a very essential protein. Even if you knock out TDP in neurons, this is lethal. So therefore, it is difficult to use an ASO strategy. Just lowering TDP may be toxic and therefore has not yet been pursued. There are some efforts, however, to try and go for an allele-specific reduction of TDP if we can design the ASO in such a way that they only reduce or mostly reduce the mutant RNA transcript, and so the mutant protein. We know that TDP has a very strong autoregulation, so if you reduce the mutant, the wild type will be increased, and in this way, it could theoretically be possible to really treat this disease, but we're not there yet. Then finally, what about sporadic ALS? If we don't know the disease cause, it's difficult to design ASOs that are silencing, for instance, the disease-causing gene. But there are several efforts underway for sporadic ALS as well, such as the ataxin 2, but also more recently stathmin-2, UNC13A, and CIF2. I will briefly talk about that as well. Ataxin 2 was discovered as a TDP modifier in 2010 by Aaron Gitler's lab. In a yeast screen, they first found that TDP aggregation can be reduced by reducing ataxin 2 expression. And later on, they showed that it also mediates TDP43 toxicity in cells and in mice. And they also identified ataxin 2 as a genetic risk factor for ALS. CAG repeats in the ataxin 2 gene typically give spinocerebellar ataxia type 2, but intermediate repeats between 27 or 29 to 32, 33 increase the risk for ALS. And so that together these data suggested that maybe when we lower ataxin 2 by an ASO, that this could be protective both for ALS and SCA2. And so a phase 1 trial, biomarker guided, was performed, again, by Biogen and Ionis. But very recently, a press release was issued, it's not yet published, these data, stating that although the ASO, again, clearly lowered ataxin 2 levels in the CSF, that it did not affect the neurofilaments nor the clinical response as measured by ALS-FRSR or FVC. And so for that reason, this program was also discontinued. Two other examples of therapies under development for sporadic ALS are stathmin-2 and UNC13A. Both are targets of TDP43. And so what happens is TDP43 normally binds long introns and can repress the expression of cryptic exons. These are sequences in the intron that should not end up in the protein sequence. But when TDP in the nucleus is reduced, this cryptic exon can end up in the mRNA. In this case, it brings with it a polyadenylation site, and so you have a truncated RNA that is non-functional, and you don't have the production of normal stathmin-2. And stathmin-2 is a microtubule binding protein that has important functions in the axon. It's important for axonal integrity and also for regrowth of axons. And so this is one of the TDP targets that could be important in the pathogenesis of ALS. To overcome this problem of missplicing, it is possible to design a splice ASO that binds to these regions to sterically hinder the inclusion of this cryptic exon in the transcript, and then can correct normal stathmin-2 transcripts and normal stathmin-2 production of protein. There are two companies working on that. One company, Curalis, has an ongoing phase 1 study. It's too early to say anything about it, but it's running, and so I think within a year or so we might know more on how effective this strategy is, first to correct the cryptic exon expression, and also at influencing neurofilament levels or clinical outcomes. UNC13A is also a very interesting target for therapy. UNC13A is a synaptic protein, also in motor neurons. It plays a role in the release of synaptic vesicles. And it also has a cryptic exon that is normally spliced out because TDP represses the expression of this exon. But when TDP is leaving the nucleus, this exon can be included. It contains a premature stop coding, and therefore the transcript can be degraded by nonsense-mediated RNA decay, resulting in lower mRNA levels and thus lower protein levels. Interestingly, it's also a genetic modifier of ALS. In a SNP, you have the at-risk allele, the CC allele, that increases your risk to get ALS. It's a susceptibility risk factor. It has a small effect. It has an odds ratio of about 1.15 or so, so a low extra risk to get ALS, but it also modifies the phenotype. People with this SNP typically have a faster disease progression, more bulbar onset, more likely to develop FTD as well. And the same strategy for therapy can be designed here as well. A splice morpholino that prevents this cryptic exon to be included in the transcript is under development and will also be a very interesting target for further study. Of course, those two last approaches, stathmin-2 and UNC13A, they try to overcome some of the downstream effects of TDP nuclear loss of function. TDP affects hundreds, if not thousands, of genes, and so the question is whether modifying one or two of these targets will be enough. But there are also other strategies that try to go even more upstream. And so this is a paper that reports on CIF2 silencing. CIF2 is a splicing factor in the spliceosome. And in a screen, they identified that if you knock down CIF2, that you can reduce TDP43 proteinopathy and increase the survival of cells. And so the idea is that by knocking down this gene, you prevent or reduce the amount of cytoplasmic TDP aggregation and enhance the levels of nuclear TDP, and therefore prevent the missplicing induced by TDP loss of function in the nucleus, but also reduce the aggregation, the toxic gain of function by cytoplasmic TDP aggregation. So that's a very interesting approach, and of course, whether this will work, it's far too early to say, but it's just an example of how observations in the lab can nowadays give rise to ideas for therapy, which can actually be translated into trials in patients quite rapidly. Finally, I want to show another approach. This is not an ASO approach, but an AAV gene therapy approach called TDP-REC. It's not yet published. It's from the group of Pietro Fratta in UCL in London. And they thought to exploit actually this cryptic exon expression as a kind of sensor of TDP pathology. So the idea is that they build a cassette and that is actually within a cryptic exon. And so when you have nuclear TDP loss of function, this cryptic exon will be expressed and the cassette that is integrated through your gene therapy as well. And this cassette contains TDP43 with a RAVER1 fusion protein. RAVER1 is also an RNA binding protein that can repress genes. So the idea is that when you have too little TDP in the nucleus, it will be sensed because your cryptic exon is expressed, and therefore TDP is produced with a RAVER1 fusion, which goes back to the nucleus and fixes the problem. So it's an interesting approach, which seems to be working in cells, but of course there's a long way before this will be a therapy in patients. But again, another example of how many molecular strategies are possible nowadays. This brings me to the conclusions of my talk. I think ASOs offer a lot of opportunities for precision medicine in ALS. Tofersen really was and is a game changer for the field. The second ASO for FUS ALS, ION 363, holds promise, and we all anxiously await the results of that trial. For C9, it seems to be more difficult. The first two efforts failed, but maybe we need other approaches that target both strands. an allele-specific ASO is, I think, a possible solution for tAR-DPP ALS, and different studies with ASO therapies for sporadic ALS are also on the way. So we are living in exciting times for therapy development in ALS. Finally, I would like to thank the teams both in the lab and in the clinic that allow me to combine basic and clinical research, and I would like to thank you for your attention.
Thank you, Philippe, for this nice presentation. Maybe some question in the audience? Yes, I'm coming up.
Thank you very much for your presentation. I'm surprised by the fact that for ALS, the ASO approach is not specific, and you can observe reduction of the wild-type allele, I would say. Mm-hmm. How do you explain that with respect to, for example, in SMA, in which you have ASO strategy working specifically to target mm-hmm. a splicing defect? Mm-hmm. So the ASO here, in the case of ALS, is not specific to the mutation? Ah.
No, you're right. So in SMA, the approach is even more specific. Right. But these diseases, you are born with one mutant and one wild-type allele, and you produce wild-type and mutant protein, and to some extent you tolerate this, or the system tolerates this, until a certain age when ALS develops. And by just reducing the levels a little bit, both wild-type and mutant, maybe cells are able to deal again with the influx of mutant protein. If you reduce the influx of mutant protein and you rely, I think, on the protein degradation capacity of cells to clean the mutant aggregating species of the protein.
I was wondering whether you knew if the Ataxin-2 trial where patients were matched on the Ataxin-2 repeat expansion, or whether this was all...
Both. So both sporadic ALS was allowed, but also there was an enrichment for patients with an intermediate repeat expansion. But nobody has seen the results, so I have no clue whether it made a difference or not. I have not seen the results.
Yeah, seemingly, since even if there would have been a positive signal in the subpopulation, probably they would not discontinue it.
Yeah.
Okay, thank you.
Thank you very much for this very beautiful review. I was wondering, but we discussed that previously, about the dose and the way of administration of ASO, because maybe there's not sufficient biodistribution in the central nervous system, and maybe some programs are stopped too early because we don't have the good doses. Okay, you nicely remember us that you have a target engagement, so it's okay. But then sometimes you stop the program because the NFL is not going down. But maybe it needs more time or higher dose. Or I would like to have you for that.
That's indeed a very difficult question. Of course, from a company point of view, I really understand it. They have had successes, and they saw in this time frame a clear reduction of neurofilaments. If they don't see it, it's a no-go. But that's more a kind of operational answer. The ASOs, they give a reduction in CSF, but we have to realize that the levels of protein you measure in CSF are not necessarily the same as you measure in tissue. And for SOT1, it has been clearly demonstrated in primates that the reduction in tissue is more pronounced than in CSF. So reduction also for tofersen, the reduction in CSF is not dramatic. It's like 34% or 25-30% reduction in SOT1 protein, but we assume that it's more in the tissue, and for Ataxin-2, I don't know. But it's a very relevant question, but I think the advantage of these kind of precision medicine approaches is that you know if you reduce it in the CSF to this level, it doesn't work. That's not necessarily the end of the story, but maybe we have to go higher, maybe we have to treat longer, but yeah.
Thank you for your fantastic talk. I will come back to, I want to come back to the C9 ASO study. And you have shown the 90 milligram group, but you haven't shown the 60 milligram. And 60 milligram was improving. So I would like to learn what is your interpretation of that. And also I want to motivate another political statement. So do you think that the company terminated also this study too early? So this combined questions.
Yeah.
Maybe I have one answer.
The 60 milligram did very well, surprisingly. So in terms of GP downregulation, it was even slightly better than the 90 milligrams, but it also increased neurofilament levels. And I didn't show it, but also there the clinical and biomarker response was not favorable. But I agree with you that stopping early has a certain risk. Nobody will ever do this study again. And so we will never know if you treat longer, that maybe there is a delayed response. I'm not so surprised that if you mess with the biology of the disease, it can induce neuroinflammation, there can be a phase of whatever kind of altered biology. We don't really know if this is transient or not, and the only way to know that would have been to run the study for a longer period of time.
I'm here. So I'm curious on your opinion on the potential value, but also the possibility of combining some of the targets, especially for TDP, where, as you nicely said, it's very complex.
Yes, and one of the problems there is that we don't know how to select the TDP targets that are biologically relevant. There's so many of them. You can look at the which that are most affected, or the ones that are expressed in motor neurons. But I don't think, imagine that the trials of the individual ASOs for statin and UNC13A are negative. I think that it's very unlikely that someone will try to combine them. Of course, if they show an effect, a logical next step would be to combine them to see if there's an additive effect. But maybe you need to combine them, even if the single ones don't show an effect. Yeah.
Thank you, Philippe, just for the matter of time. I have just a very quick final question. Did anybody measure the C9 levels in these patients? Because you could explain potentially, I mean, if you reduce the C9 level, might trigger, as you said, all the potential inflammatory
I think they were normal.
They were normal?
But also there, it's a different story per cell type. For instance, if you look at single nucleotide sequencing data, just the effect of the mutation on C9 expression levels is different per cell type. So yeah, also if you add an ASO, I think we don't know the real answer, because then you would have to check the tissue at the single cell level.
Thank you.
Thank you, Philippe.